Welcome everyone, and thank you for joining us. I'm Brenda Dettinger from BIOPAC, and I will be your moderator on today's webinar. Today we're going to be talking about high density whole head through the hair fNIRS with EEG. It's a very full presentation, so I'm going to get right into the housekeeping notes. Please submit your questions and comments to the GoToWebinar control panel. That's how you communicate with me, your moderator. When the webcam is turned on, if it looks small to you, you can grab this little gray bar here and resize it. Today's webinar is being recorded and we'll send a link when it's processed. Finally, we have a survey at the end of the webinar. Please complete that so we have your feedback and ideas. Now, before we get to our presenters, I have an announcement. Our biennial training conference has a new name. We are now the 4T-Phys Human Physiology Conference, also known as 4T-Phys Training, and conference registration is open. This is an in-person event on July 17th here in Santa Barbara, California, where we're based. We will host 3 days of workshops on physiology, data recording, and analysis. I just want to show you the agenda real quick. We have a great agenda, including a new fundamentals track that will be organized by small groups so you can do hands-on learning for all the major signals. We have a new virtual reality track as well. This is our third time doing this conference, and we'd love to see you in person in July. All of our staff will be there, our reps from all over the world, and we'll have many of our partners, including today's presenter, Thomas from Seenel. Now on to our presenters. I'm excited to introduce our experts today. We have Thomas Fontaine. He's the CEO and founder of Seenel Imaging. Prior to Seenel, Thomas spent a large part of his career working in leading worldwide medtech companies such as Medtronic and Covidien. He held several positions in France and EMEA. Seenel is a spinoff of an INSERM University Hospital laboratory, and it was founded in 2018. Welcome, Thomas.
Welcome. Thank you, Brenda.
We also have Dr. Emmeline Moulier. She obtained her PhD in 2020 at Lausanne University Hospital in Switzerland. During her thesis, her research focused on linking structural and functional MRI connectivity on patients with neuropsychiatric disorders. She joined Seenel Imaging as a neuroscience application specialist in 2021, and she works on combining electrophysiological and hemodynamic responses in the new neuroimaging device for EEG and NIRS. Welcome, Dr. Moulier.
Thank you, Brenda, for the presentation, and welcome everybody.
We also have Fraser Finlay, the CEO of BIOPAC. He has over 20 years of experience in the life sciences and physiology monitoring industry. Fraser oversees the development of physiology, data collection, and analysis products, and is a well-regarded expert who presents at workshops all over the world. Welcome, Fraser.
Welcome, everyone. Thank you, Brenda.
All right, Fraser, let's start with you. I'm making you presenter.
Perfect. Okay, thanks, Brenda. I appreciate it. Good morning, everyone. Welcome to our webinar. Organized a little bit here, move my video out the way. All right. So before we start this morning, I have a little background information about BIOPAC. BIOPAC develops high-quality scientific tools that allows researchers to measure physiology anytime, anywhere, with any subject. We've been in business for over 30 years, and 99% of the top universities use BIOPAC products. And those customers have now generated in excess of 48,000 citations. Righty. So I'm delighted about this new partnership with Seenel Imaging because it allows BIOPAC to expand its fNIRS options for our research customers. The Seenel Imaging product line is called MedelOpt, and Thomas will provide details about the different options during his portion of the presentation. The MedelOpt whole head solution provides users with the opportunity to record data from different areas of the brain and combine the the hemodynamic signals with the electrical signals EEG. The unique exoskeletal design of the headset allows users to access the sensor site and easily remove hair to make setup easier and ensure high quality data. And Thomas will provide a really nice demonstration of what I'm talking about there. The headset also adjusts to accommodate a range of head sizes, from children as young as 4 years of age to adults, to meet the demands of your study population. This means that you don't need specific caps for different head sizes, basically one size fits all. The MedelOpt family interfaces with BIOPAC data acquisition systems and stimulus presentation options, plus third-party devices to provide a complete solution for researchers. So I'm going to hand back over to Brenda quickly, and then we'll learn a lot more about the devices from Thomas and Emmeline. So Brenda, back over to you.
Hi, that was fast. Okay, here we go, our first poll in the first 9 minutes. This is kind of a two-part question here. So we want to know where you're going to be collecting data from, but also are you using single subjects or dyads in your experiments? And you can select multiple items there. Lots of answers coming in. And then if you missed my my intro in the beginning, just so everybody knows, you can you're welcome to pass questions to me through the questions pane in the GoToWebinar control panel, and I'll ask them whenever I can. Sometimes I ask them during polls, or I'll save them till the end. I'll get to as many questions as I can. Okay, so let's see here. We still got some answers coming in. Yep, looking. Most people are in the lab. Over 85% of people are in the lab. About a third in the field. About a third in the field while mobile. And then singles versus dyads, we've got about 55% singles and about 22% dyads. So well done. Thank you guys for responding to the poll. I'm going to go ahead and close the poll. And now we are moving on to Thomas. I'm making you presenter. Here we go. I see your webcam. Looks good.
Yep. It's coming. So that's perfect. Thank you, Brenda. I appreciate it. Good morning, good afternoon, good evening for everyone. So I'm Thomas, as shortly introduced. I'm CEO of Seenel Imaging. I'm really excited today to present to you our research device, so-called MedelOpt. And thanks, Fraser, for the short introduction. So before I jump in my presentation, let me quickly show you how the device looks like in reality. As you can see here, hopefully, we have indeed developed a device by researchers for researchers. And MedelOpt is actually a wearable research device combining EEG and high-density fNIRS device. So MedelOpt get easy access to the scalp, and this is one of the key features of the product, and we'll see that during the next coming minutes. And you will understand why it's so important to get access, especially for fNIRS reason. And third, I would say that MedelOpt offer a lot of flexibility and modularity, I mean, to meet any of the research requirement and expectation as well. So it's indeed easy to fit any mapping and to address any region of interest on the cortex. So let me briefly show you how does it work, and then I will jump in my presentation. So as you can see here, we have the headset with the exoskeleton, which is a unique design for fNIRS and EEG. One of the key topics is indeed I already introduced on top of the wearability is to make sure that we can direct access to the scalp, and this is something that we can probably do here. As you can see, I can remove dioptodes and I can then move any of the hair below each of the otone, which is critical with regards to the signal's quality, the optical signal's quality. One of the other things that we can address with the device is that we can change the setting, and you then can customize the setup of your device the way you need to really address the different region of interest while doing the acquisition. So as you can see here, I'm having a short arch, but I'm also having longer arch to address different region, and I can do whatever I want. So I can bring all the arch back frontward or backward the way you need in order to make sure you can address anything you need. So if I'm going to in details, let's see how does it work with an arch. So as you can see here, I'm having an arch which is actually full of holders where you can place some sensors. So what we can do is actually, I'm going to show you an LED, so the red, let's say red flag sensor. So it's a dual wavelength LEDs that you can plug in any of the sensor location. We have also the photodiodes, which is the blue sensors. So it's preamplified silicate photodiodes that you can also plug in any of the sensor holder. And last but not least, as I introducing to you, we have the green sensor, which is actually the EEG. It's a AGCL electrodes. Then you can place as well on any sensors on the device by itself. So as you can see here on the device, we have the different product, and I then can change easily the way that I'm placing any of the sensors on the system. So you can really customize your mapping the way you need, the way you want. So you can really change the way, and then you can move, you can slide indeed the location of the sensor holder the way you need. So this is the way that we are doing the design of the exoskeleton, and I'm really excited today to present you one new recent development that we have addressed is that we have integrated this arch and all these sensors on a new device, which is actually a virtual reality headset. So as you can see with this Quest 2 system, but it could fit to any let's say hand-mounted device, we can really address any of the sensors, and then I can wear it fairly easily as we are having the, let's say, the all the electronic entity on the back of the entity, and then you just have to click, and as this is a wireless version, you're ready to go for your protocol. So I won't go into much detail in that, but that means that this is really a new development, and very excited to propose this development as this is now ready, and then you can either use our MedelOpt system for any research application as well as for let's say virtual reality. Let me share my screen now.
Hey, Tom, yep, you got it. There you go.
Thank you. So as you can see on the screen, I'm just jumping in a very brief explanation on the near-infrared spectroscopy. So I'm talking about fNIRS, which stands for Functional Near-Infrared Spectroscopy. So actually similar to fMRI, fNIRS detects the changes of hemoglobin chromophore inside the brain, but via differences in optical absorption. Whereas most of the biological tissues are transparent to near-infrared light, oxyhemoglobin, which stands for HBO, and deoxyhemoglobin for HBr. This is the most common acronym we're using. Both are known to absorb or scatter the near-infrared light and of slightly different wavelengths in the range of 700 to 1000 nanometers. So light emitters are placed on the surface, as you can see here, on place of the surface of the scalp, radiates near-infrared light into the head. Given the differential absorption and backscattering of the oxy and deoxyhemoglobin, a portion of this near-infrared light return to the surface, of the scalp, where it is being measured by photodiodes, photodetectors. So, shall we move ahead in the presentation? fNIRS provide an indirect measure of the localized brain activity. The modified Beer-Lambert law is one of the most used algorithms to convert the measured light intensity into a concentration change of HBO, HBr, oxy and deoxyhemoglobin. So on the right-hand side of the slide, you can see the typical hemodynamic response. I'm coming back to the fNIRS and EEG and the complementary of both modalities. So light scattering limits fNIRS signals to the outer of 2.5 centimeters of the brain with a spatial resolution lower than fMRI, but higher than EEG. And although somewhat limited by the time lag of the blood flow responses, which occur on a scale of seconds rather than on millisecond scale of neural impulses, fNIRS offer a much better temporal resolution than fMRI. Combining fNIRS with EEG can give you information both about spatial and temporal aspect of the neural activity. So let me briefly review the fNIRS limits and benefits, starting with the benefits. As you probably understood, there is a good to medium spatial resolution provided by the fNIRS. We are measuring separately oxy and deoxyhemoglobin, and we can integrate different modalities, as you saw earlier today, the virtual reality, but could be any other kind of modality, any kind of BIOPAC physiology sensors, I mean. We can also use this device in ecological context, as this is there is a wearable option. And the device, as shortly introducing by Fraser, can be either used on a child from the age of 4 until adults with absolutely no issue, and we're using the same headsets. One of the limits that you need to consider is the depth, the penetration depth of the light, which is going up to down to 30 millimeters. So that means that you're just acquiring acquisition on the cortical activity. And one of the key points which makes indeed our device unique on the market is about health. Hair remains the natural barrier that affecting the most the optical signals quality. And most of the devices available on the market nowadays are using EEG cap based device, I mean. So that means as soon as you're wearing the EEG cap with the fNIRS on the head, you don't have much access to the skin scalp. So you have in between the sensor and the skin, you have the hair which providing the natural barrier. And this is extremely important while looking at acquisition on hair results. So that being said, let me come back now to MedelOpt. So MedelOpt can be used in many different research applications, from let's say BCI to sports science to now virtual reality, or even hyperscanning as well, language and so forth. So as you understood, MedelOpt is a bimodal device, and we are, as long as I know, the only device which is fully integrated EEG and fNIRS. I mean, we have integrated, of course, both sensors, the ones from the fNIRS, the one from the EEG on the headset, but it's also, as you can see on the background of the slide, we have also integrated those modality on the same software, offering the shortest latency time for both modalities down to 2 milliseconds. EEG, it's a system of 8 AGL electrodes with gel, and we are using continuous waves fNIRS modality. While looking at the direct access to the scalp, as you probably understood, and we'll see that in our demonstration later on how this affects the signal's quality. As you can see, I can really remove easily any of the optodes, and with a little hook, I can move away any of the hair below those optodes. So this increases the signal's quality, even can get some new channels where blocked, I mean, by the hair. With regards to flexi-modularity, as you probably understood earlier, one size fits all, as we can manage with the same headset different size from child to adult. We can also manage multiple brain region. As you can see with the same headset, with the same sensor on the headset, we can either choose to place them on the frontal, motor cortex, occipital, or we can spread all over the head depending on the density. And this brings me to one of the last features of the product, is that the device could either be used with or without EEG. So you choose if you want to get EEG within the device or not. I mean, if you just invest in the device and the next day or the next year you want to get EEG, you just need them to buy electrodes. All the amplifier remains within the device by itself, so just upgrade it by having some new EEG headset. It's the same principle with the fNIRS optodes. I mean, we are offering different density of fNIRS, from high density, which is what we call the version 8 and 8 emitters, 8 receivers, as you can see on the left-hand side of the slide. And you can choose to either use with up to very high density, which is the highest density we're proposing in fNIRS, the 16 emitters and 32 detectors on the headset. So you can progressively, once if you need to add some additional optodes on the headset, by the time if you want to get some additional region of interest. I mean, if you choose to do one region of interest like prefrontal, an 8 and 8 version is really good enough. And if you want then to move towards over over region of interest, it might be wise indeed to get some additional optodes as well. This also addresses one of the key points, which could be the financial constraint, as indeed the more you get, the higher is the price, of course. So last but not least is about wearability. This is a wearable system. We can offer it in a wireless version. So that means this is an endless range of acquisition. So you can either choose to use it while doing, let's say, exercise, walking, cycling, being in a, I mean, in the car or in a plane. Absolutely no issue with our device. So I guess this is time now to go towards the poll. So Brenda, I think this is up to you.
Great. Yes, I'm going to go ahead and launch the second poll. Let us know what the most interesting feature is now that you've seen that quick demo of the device, the physicality of the device. Okay, so I have one quick question for you from Julian. What is the sampling rate while we're waiting for the poll results to come in?
Yeah, so sampling rate is coming from 4 Hertz to 32 Hertz for the fNIRS. So in the demo that we are showing today, the fNIRS system runs at 16 Hertz. And for the EEG, we are having a sampling rate of 512 Hertz for EEG.
All right, so I'll go ahead and close this poll. Wait, a couple more results coming in. I'm going to go another 5 seconds. If you haven't responded to the poll, please do. Lots of other questions coming in. I see that from the audience, so we'll just try to answer one during the next poll, and then we'll save the rest for the end. Okay, so I'm just going to share with you the results of the poll, and then I'll tell you my favorite feature, okay? So first of all, the most popular feature is the combined EEG and fNIRS. followed by wireless mobility, optodes, montage, flexibility, and access to scalp. So thank you all for participating. I personally love the flexible modality and being able to add on depending on your needs for each experiment. So, so if it was me, that's what I would answer, but I didn't answer the poll. All right, so now we are moving on back to Thomas, right?
Yeah.
Okay, go ahead and share your screen when you're ready.
Yeah, sure. I will. Just give me a couple of sec. So I know I'm sharing my screen with a short video showing how the system works, how we can set up on the head, and how it's running up to the software. So
Second, your screen is paused or something. So what are you supposed to be showing a webinar screen? It's kind of stuck between poll and webinar.
Okay, let me share.
Try that again.
Yeah, it should be working now.
Now I see an agenda slide with the blue background and setup of MedelOpt headset on a participant. Yes.
Okay, right. That's perfect. So let me, I briefly introduce, we are using today, and this is what we're going to propose for the whole demo that we are showing you today, a version of MedelOpt, which is 816 EEG electrodes. So 8 stands for 8 emitters, 16 for 16 receivers, and EEG. So as you can see on the right-hand side of the screen, we have designed the montage to address two regions of interest. So the prefrontal part for the frontal, the prefrontal part in red with some fNIRS devices, including the short channel, and in yellow the motor part, the motor cortex part, with some over. So this is a standard way to make MedelOpt, but again, we can increase the density if needed. So let me keep going on how the system works. So you will see that in a sec. So we're going to start then as soon as, in any EEG system, you're going to have then to start by measuring, the Gaussian to Union to make sure that you can well place the headset on the head. So it's very similar to what you are doing, I guess, on a day-to-day basis for EEG. And then you're going to start to set up the EEG when it's ready. So first things you need to set up the headset on the head of the participant. Of course, we can, as you may have seen, and thanks Emlyn for being the participant today, we can have participants with glasses. This is absolutely no issue for us, thanks to the specific design. And then we can start to tighten the exoskeleton headset on the head to make sure it does not move at any given point of time during the acquisition, which is absolutely key. So I have today I have 6 positions to tighten, so 2 in the front from the new strap, and then we have some in the back of the acquisition where we have our electronic box and all the cables. So this is where we can adjust the system. I'm doing the same way for both and right, of course. And then the next step would be for me to tighten the straps of of the headset. So as you can see on the screen, to make sure that any of our optodes are well placed and in contact with the skin or actually with the hair to start. So just making sure that you have a well placement and everything runs well. So usually it's depending on the time you need, but it should not take that long to make it happen, especially as we are, as you may have understood, we are using a pre-configured headset. I mean, every of the optodes and the electrodes are already in place on the system. So you just have to, let's say, plug and play to tighten the headset, and then you're ready to go for the next steps. So we're gonna see that in a few seconds for the EEG placement. So like you're doing, I mean, for any EEG setup. We just use a gel, so it's not a dry electrode, it's gel-based electrodes. So we are placing some gel on the 8 electrodes. So once we do that to improve the impedance, and we'll see how we measure the impedance on the software stuff. Don't forget indeed to make the reference with the hair clip. We'll then now ready to start for the fNIRS setup. So as for the fNIRS, as you understood, the first thing to do as soon as you have some hair and some optos on the hair is to make sure that you can remove the hair again, and this will increase the light intensity and the signals quality of your optos. So you just need to remove, let's say, the optodes and to move away the hair on the dedicated resources. In our example, we do have to do it on any of the optodes on the motor cortex part, but we're going to have to do as well, as there is long hair, on the prefrontal part, making sure that we are not having any issue with the hairs while doing the position. So by doing so, you're really increasing the signals quality, and this is, let's say, as the way that you set up the system on the head, this is a key part of making sure you can get some high quality signals by the setup. So let's go then now towards the software. So once you have connected the software, to your setup, we just need to switch on the amplifier, and then we just start to measure the impedance of the 8 electrodes. So as you can see on the right, in the middle of the screen, you have the impedance measure for each of the electrodes. So when you see the numbers are being reduced, it's because I'm applying some gel below the electrodes. So the ambition is to reach from 5 to 10 kilo ohm to achieve the good signal to noise ratio. So of course, it's a bit short now with the video, but usually not take that long to make it happen. One of the key interesting part, of course, is when you're starting to make and to use the fNIRS. So as you can see, I'm going to stop a bit the video just to explain. So on the top part of the slide, you can see the 1 to 32 photodiodes representing in the graph. In the lower part of the slide, you can see the numbers 1 to 16, which is actually the LEDs. And this is where you're going to then check how the LEDs are affecting the light intensity on the photodiodes. So let me keep going. As you can see here, I do not have any of the LED running because we are at 0% of the light intensity. So the only thing we are measuring, as you can see, as we are receiving lights here, it's only the external environmental light that is being received because the headset is placed on Hemelin heads, and we're receiving lights from external lights. So what we're going to do first, and you will see, we just place a blackout cap on the head to protect any of our optodes from the outer light, and you can see that we're reducing to 0 the photodiode. So that means any of the optodes are receiving lights today. So again, as this is a 8 to 16 photodiodes system, only the 1 to 16 photodiodes will run today. The other one are not connected. So the next step, and this is where we are checking how we are having some good signals quality, we can then click on the LED 2, the sources 2, and we can progressively increase the light intensity. So on the bottom of the slide, you can see that we are increasing the light intensity. So in this example with the LED 6, we can increase it to 25% in infrared, and we can see which are the closest photodiodes receiving some light. So this gives you a good understanding about the light quality and if it's good enough. So it's fairly easy to make it happen as soon as this is on the prefrontal part, because our optodes, either the sources or the receiver, are directly placed on the skin. And then it's a different topic while you're achieving, let's say, the hairy zone. So in the next sensor, we have removed the hair, and we can control how much light we're receiving from the LED 8. So you can see that the closest photodiode 9, 10, and 11 are receiving some light, which is very different from, let's say, the photodiode 10. I hope you can see that, but we have increased the light intensity to 80%, and none of the photodiodes which are close to the LED 10 are receiving any light. This is mainly due to the fact that we are having hair below, in between, let's say, the scalp and the photodiode or the LED. So what we're going to do, as you probably already understood, we just need to remove the hair as we are doing initially. So we just remove the hair, and then we can do the same exercise and see how much light we can now receive. So you can see that by having 15% of light intensity, actually we are providing a good signal. So if we are moving up the light intensity to a higher ratio, which is going up to 50%, you can see that our photodiodes are receiving some light. So this gives you some more visibility how the light is being blocked by the hair. So once you're ready to go, you're happy with your system, then you just need to calibrate. So the system will launch an automatic auto calibration, and then you will be ready to go for the recording acquisition. So you will go to the monitor page, and you will achieve now this page, and you can see the EEG on the top of the slide. So this is where we can see the 8 EEG electrodes running, and on the bottom you can see today in this presentation only one channel, which is the channel source 2 to detector 2. It's a real-time HBO HBR. So some few things that you can do as a starting point is to check if you can see the eye blinking, asking your participant to eye blink, I mean, as a starting point. So you can see that on the frontal, let's say, electrode, and you can also notice the alpha waves on the electrode placed on the occipital part while asking your participant to close the eyes. So let me keep going. The first test would be just to make, like usual, I guess, jaw contraction, and you can see now we're doing the jaw contraction, a lot of, let's say, artifact. So we are sure to get good to signals ratio for the EEG. And now let's zoom a bit on what happened on the heartbeat. So I mean on the HBO HBR. So as you can see, we're measuring the heartbeat. We can see the heartbeat in the cortical activity as we are running at 16 Hertz in our example, and we can measure real time what's going on. So what we are doing, of course, by doing this kind of measurement and real time measurement, you can check during the acquisition if the signals, either EEG or HBO HBR, are the right one, and you can then progress. Or of course, it's fairly easy to change either the channels. You can choose which one you want to get. You can see up to 4 different channels at the same time, maybe for 4 different regions if you need. So it's something that you can do fairly easy. So you have different setup. I won't go into much detail today, but I will be happy to answer any of your questions in the long run or after the webinar. So once we have performed our acquisition, the last step would be for us to provide you some raw data, which are available, I mean, in text file format or in a SNIRS format, which is a standard fNIRS format nowadays, or in a .edf as well. And I guess Fraser will show us today later on how we can post-process some EEG data from MedelOpt in the Hacknowledge BIOPAC software, and SNIRS usually, so it's for the fNIRS modality, could be then post-processed, I mean, on any Python or MATLAB software, open source software, I would mean, as we can post-process those data either on Home F3, which is one of the most common open source software, or Nearstorm or SPM. There is some few, so you can use the way you want. So this is it from my side. I hope that it is clear for you, and I guess we can go to the next presenter, but I think there's a new poll now. So I guess this is it from my side.
We have our final poll of the day. I'll go ahead and launch that poll. Which areas of the brain are you interested in? And I do have a couple questions here. I will probably only ask one during the poll. Gosh, lots of questions coming in, you guys. Very active audience. I love it. All right, an easy question to answer during the poll. Sometimes we don't want to get too complicated during these, but Elizabeth asked, how is the wireless data transferred?
So actually, we are transferring the data. So we are using a Wi-Fi system with what we call a mini PC, which is directly linked to the headset. I mean, you can put that in your pocket, and then we can record the data in that small mini PC, and then you can get those data from the mini PC through server or through let's say disk or whatsoever. You can use it in a different direction. So you can either choose to see real time on the screen, or you can see them afterwards when you have transferred the data.
Okay, great. I'll go ahead and close the poll. It's almost tied between prefrontal cortex and whole head, and the rest of them are kind of equally matched. So great. So now it's time to go to Dr. Moulier. Here we go. I'm making you presenter.
Okay, hi everybody. So now what I'm gonna do is to show you how you can do a complete acquisition from the simple protocol to the post-processing part. So the protocol we're gonna use today is a simple hand-grasping protocol, which is often used as a validation step in NIRS. Then I'm gonna show you a bit of how you can load the data and visualize your data and post-process them quickly with a simple pipeline, and how you can project this activity back on the brain, which is at the end what we're interested in as researchers. So first of all, let me just remind you, I'm using the same montage as Thomas, which was with 8 sources and 16 detectors, covering frontal and motor part, because we're doing on-grasping, so we're interested in the motor part. So NIRS protocol are usually block-based, like in EEG, but the timeline is a bit different because the hemodynamic response is much slower, and it takes a few seconds for the oxygenated signal to increase and decrease and to see this variation in concentration. That's why for this protocol, we're gonna do 30 seconds of baseline and 10 blocks of 20 seconds of grasping, the time that the oxygenated hemoglobin can increase. And we are gonna follow it every block by a resting time to let the time to the variation of oxygenation to go back to baseline. So as you can see, I'm still the participant. So I'm doing this 10 block of ungrasping. I have on the screen a visual cue telling me when to grasp, and for resting time, I have a cross I have to stare at. As Thomas showed earlier on the software, you can see the EEG in real time. You are going to be able to see the trigger here on the auxiliary signals, and you can select up to 4 NIRS channels that you can see in real time, and you can select them according to your montage. As Thomas mentioned, when all the recording is done, we are going to have the data in text or NIRS file. And I'm going to show you how you can open directly and visualize your data, which are in NIRS file, using one of the most famous toolbox for NIRS, which is Homertree, which is MATLAB based, but it also exists a standalone version that you can use even if you don't have a MATLAB license. So this is how it looks like. On the right, what you can see is your montage that you design, and on the left will be the window where you are going to have the signal displayed. So if I click on source 1, which is in red, I can see all the signals, all the channel involving source 1. The color helps us to differentiate the different channels. So in purple, I have the source 1 detector 3 channel I can visualize. in pink, I have source 1, detector 2, etc., etc. The color will help us to see which channel we are visualizing. Here, I'm visualizing the raw data at one wavelength, 660 nanometer, but I can also switch to the other wavelength, which is the infrared one. And I can also switch off detectors or source or channel I'm actually looking at. So in Homer, you have a lot of post-processing options, but if you just click on run, you are gonna run the default post-processing pipeline, which is pretty basic, but it's a good pipeline to start with. So in this pipeline, you will do a basic reconstruction from raw data to oxygenated hemoglobin reconstruction, basic filtering, and the average of your block, because as I said earlier, it's a block-based protocol. Once it's run, you can now visualize different steps of post-processing, so optical density and now concentration. So what I see here, it's still the source 2 detector 7 in green channel, but what I see now is the HBO signal, so oxygenated hemoglobin and not the raw data. I can add to it the deoxygenated signal, which appears in a dash line, and plain line I have HBO. And I can see all my signal over all my acquisition. The vertical bars you can see are actually my events and stimuli mark. So if you have a trigger, it will be automatically detected and put in your NIRS file. Here I have 3 types of events. So my blue marks are the start of baseline, my pink one is the beginning of ungrasping, and my yellow are the stop of ungrasping period. This event and stim mark will be the one which are going to be used to compute the average of the block. So Homer can automatically compute it, and it's what it's called hemodynamic response function in Homer. And what you can see here will be for source 5, or in source 4 now, the average hemodynamic response for the condition baseline. So I can see, so this is the average. So at 0, this is the beginning of all my stim mark baseline. I can see from -2 to 20 seconds after the stimulus. So the average over all my blocks. I can easily edit in the option and post-processing step of Homer to increase, for example, this time window. So here I change, I want to say, okay, I just don't want to watch 20 seconds after, but I want to see up to 30 seconds. So this is what I do here. And it went pretty fast, but here I change also the condition I was visualizing. So now I'm able to visualize the block average over my 10 blocks of hand grasping from my motor channels here on the left hemisphere. And I see from -2 seconds to 30 seconds after the beginning of my hand grasping. So I'm on the motor part here. I'm visualizing 3 channels. I can see that before I start hand grasping, I have HBO, which is really close to baseline, and a few seconds after, I start seeing a variation in my concentration of HBO increasing. And after the 20 seconds, which is the end of my stimulus period, I start seeing the decrease, and I'm going back towards baseline. If I check now a similar channel, but in the frontal cortex, which doesn't have to be necessarily activated in the hand grasping, I don't see this peak of HBO. So we saw like different channels, but also Homer allows us to make us possible to see it for the entire channel at the same time in respect to the geometry of the probe and the montage. So this is what we can see here. So this is a first plot, so after you can tune it and play with the scale. But we can see it's low activity in frontal and a bit more of activity in the motor area. And we are still with the condition hand grasping. What we can do is change the condition to compare to what happened during our block of baseline. So it's a noisy channel, but in the block of baseline, we can see that we have less activity. So I played a bit more with the post-processing. And what we can see, I took just 2 screenshots of these 2 conditions, and we can see a bit more like the difference of activity in the motor cortex while on grasping and during the baseline. This is a quick overview of which type of response you can have with HOMER pretty easily. You can also tune and change your post-processing, but this is a type of activity you can expect to have fairly quickly after your acquisition. The next step will be to project this scalp activity onto the brain. And for this, there is a tool. I mean, there are several tools, but the one we're using is Atlas Viewer, which is also developed by HOMER team. It will allow us to do the registration of our probe on the brain template, a head template, as we can see here. So we are able to visualize our montage in 3D on the head template and the different channels. There is also a function, as you're gonna see, which is gonna map this back onto the brain. So you can actually do the registration and have for each optot the corresponding MNI or lab name on the cortex for your analysis. And when this registration is done, you are gonna be able to plot any HBO concentration we saw earlier on in the scalp domain with the shape of a brain map. So here, the probe I showed earlier with the hand grasping, I can project it with a color map on Atlas Viewer. And this is what it's gonna give me. So I can see it on the brain this time that I have less activity on the frontal cortex, while on the motor part, I will have more activity during the hand grasping session. So Atlas Viewer will allows you to plot all this average hemodynamic response for different conditions for all your participants, and it's going to be the basis for your analysis according to your research question. So this is an grasping condition, and after we just have the same type of plot with the same color map, but for the baseline, and we can see that, we'll see it in a sec, that for the baseline, actually, the CART is more homogeneous, and we don't have like high activation or HBO at rest, like we are really in a more baseline resting condition. So this was to give you a really brief overview of what you are going to be able to do, from which type of protocol you can do to which type of plot or signal you can expect. And I was pretty fast, but I would be happy to answer any of questions later. And now I'm gonna let the hand back to Fraser, who is gonna show you a bit more about the EEG part of the data.
Okay, I've handed back to Fraser.
Thanks, guys. Thank you very much, Emmeline, for that great presentation. I'll turn my camera on. There we go. Thank you. Okay, so I wanted to talk a little bit about the integration that BIOPAC can offer. So, many of you are interested in multimodal type setup. So combining the fNIRS with EEG is one nice step. But then if you want to combine other physiological signals, all of this can be synchronized through triggers that can be sent to the devices, and then any physiological data that's collected through, say, a BIOPAC MP160 system, for example, can then be synchronized with the EEG and fNIRS signals that are coming in. And that applies to any of our wireless or tethered C-series or D-series amplifiers. There's no restrictions there. In terms of performing the actual synchronization, looking at the image from left to right, on the left-hand side there we've got the STP100, and using the STP-IO we can interface the trigger device, the MedelOpt trigger cable, between the STP, which is connected to an MP160 system, and then on the other side it plugs into the rear of the unit on the MedelOpt headset. There is also a wireless option as well, so those of you that are doing mobile applications, you can also do the same thing, but then your participant has the freedom to be moving around and you're still able to put those event marks in. Bringing in the data from the devices, Thomas gave some examples. As far as the EEG is concerned, you've got text, .mat, and EDF file formats, and Acknowledge supports all of those, and they come in really easily, and I'll show you that in a moment, give you an example. But basically, the image that you're looking at is the data that was imported from an example file. And I'm going to use that for a little demonstration. So I have Acknowledge set up here. And if you look at this screen, you can see I'm using focus areas, and I've already marked these. And each of the focus areas represents a task that the participant was performing. And if you remember early on, we were talking about eyes open versus eyes closed. So this just marks segment number 1, where the participant had their eyes open, and then they had their eyes closed, and then section 2, eyes open again, and then eyes closed. And we'll use that to just do some quick analyses. But in terms of bringing the data in from MedelOpt, basically go to the Open menu, and down here our software interfaces defaults to the acknowledged file format .acq. But if you click on this option down here, you can see there's a number of different options. One of them is .mat, the other one is EDF, and text is right here. So those are the choices that relate to the MedelOpt software. I had already brought this in and saved it as a .acq file, and that's what we're looking at here, but pretty straightforward to open it up and bring it in. Now, in terms of analysis, there's some nice tools within Acknowledge. If you come down to the analysis menu, and there's a sub-menu here giving you options for what I would describe as the more typical types of analyses. Delta power, derive alpha, RMS. You can filter multiple channels at once, derive their frequency bands, so alpha, beta, theta, delta, gamma. You can do an EEG frequency analysis over different epochs, and you can determine the different epochs. You can remove EOG artifacts, so if you've got a lot of blink in there, you can do that. Seizure doesn't really apply here. Okay, so these are some different options. You've got all the usual tools for taking measurements at different items. What I've done is I've created a couple of workflows that will just speed this up a little bit. So for those of you unfamiliar with workflow allows you to automate repetitive tasks, so you don't have to keep going back in there and doing the same thing over and over. And you can apply workflows to a whole number of files, so you can batch process things. So I'm just going to open up a couple of ones that I've made. The first one is something that many of you will probably want to do if you want to come in and condition your data. So what I'm doing is I just set this to run a comb band stop filter, and this will eliminate any 50 or 60 hertz noise. In the example file, this was collected in France, line frequency there 50 hertz. So I've got this set to repeat over the 8 channels, and then so I'm looping this 8 times, and then I'm just running the frequency bands analysis just over channels 7 and 8. Very easy to run. Just hit play. In the background, you'll see different things happening, including a whole number of channels being added. All right, so I'm gonna minimize this for the moment. And just to make life a little easier for everyone, I'm gonna shut these files, these channels down. So what these are the different frequency bands, so alpha, beta, theta, etc. This one here is the alpha for channel 7, and I'll turn alpha on for channel 8. And I'm just going to auto scale these waveforms. So now as we're looking at this data, and actually I'll turn some of these channels off, channel 1, 2, 3, and 4, make it a little bit easier on the eye. So channel 7 and channel 8 alpha relates to the alpha wave activity from the blue channel and the alpha wave activity from the green channel. And you can see now where I've applied these focus areas over the different regions. And you can see during eyes closed, we are seeing an increase in the alpha wave activity, diminishes when the participant closes their eyes, and then increases back up again. So just to confirm that, if I come back into the workflow editor, I'm going to open a new workflow. This time, I'm just going to do a frequency analysis across the waveform. So I've just selected to do a 10-second epoch just over channel 8, which is the very bottom channel. And I'm going to look at the focus areas specifically. And if I run this, take a couple of seconds while it's running. And you can set this up to do it across many channels. You can increase or decrease the epoch size as well. Just depends on what your protocol demands. I'm just using this as a show and tell as much as anything. Now in a moment, Excel will open up, and it just opened up on my other monitor. If I come over here, if you look along the bottom, you can see that we've got 4 different tabs. The first one is eyes open, and we can see the medium frequency is 1.25 approximately, and approximately 4 hertz. If I come to eyes closed, you can see that we're right in the alpha band. Eyes closed, it's dropping back down again, and then the last segment, eyes open. So it gives you an idea of how easy it is to bring the data in from the MedelOpt device. You can perform the analysis. This can also be brought in with other signals. So if you're collecting electrodermal activity, respiration, ECG, all of these can be combined together by using event marks from the stimulus presentation system, whether that be BIOPACs or a third-party Superlab ePrime. Everything can be tightly synchronized around those events. Anyway, this gives you a really good overview. I think Thomas and Emmeline gave a really thorough and detailed presentation of the fNIRS system. Hopefully, I was able to provide a little bit of added as far as bringing the data into acknowledge. At this point, I think we're ready to hand back over to Brenda for some questions.
Yes, we are. Before we get to the questions, I just want to let everyone know, here, let me show my screen real quick, hang on. If you missed it at the beginning, our 4T conference registration is open. It's a new name with a lot of great content. We've done this conference 2 years in the past, and it's very well regarded from a training perspective. We'd love to have you here. If you can make your way to California, it's a great experience to do the hands-on training that we're offering with this device and with other devices. You know, all the staff will be there. We'll have a lot of hands-on training. We'll have other third-party partners there as well. Thomas will be there. I'll just show you the agenda real quick. I know I showed it at the beginning. It's 3 full days of sessions. Please visit our website to check it out. Early bird registrations ends February 28th. All right, now on to our questions. Okay, here we go. There's a lot of questions. I don't know if we're gonna get to them all. We do send a document at the end in the next few weeks. We'll compile all the questions together and then give you answers. So stay tuned for that. Okay, Peralm asked, for EEG, should gel be used? Is there a need to disassemble the device before cleaning? Can it be completely immersed in water?
So I'll take it. So we can't indeed immerse the full headset today into the water. We just have to remove, the electrodes out of the system, which is very easy, and then you just have to clean it. So only with a few minutes you can remove the electrodes to clean it between participants.
That, what is the total possible number of electrodes from Andre?
So we have today 8 EEG electrodes, and we don't have any other on top of the ground and reference, of course. And it's a different scenario for the optodes, which is the optical sensor for the fNIRS, which is starting from 8 emitters, 8 receivers, up to very high density, which is 16 emitters and 32 receivers. So it's always 8. It's coming by a bundle of 8. So you can go in between with any kind of version.
Right. Next question, Zayan asked, is the short channel regression implemented to this data processing pipeline?
Yes, so I guess I will take it. So we have the possibility to have some short channel to the device, and in Homer you have a function which is called GLM, which allows you to regress the short channel. So you can also choose if you want to regress a closer short channel or which short channel specifically you would like to regress.
Okay, I actually have a question. I very rarely have questions for myself. So Amy asks, will the team be sharing this webinar file with us in the PowerPoint? So yes, we have recorded the webinar. We will email a link out to the recording, and also we'll share the PowerPoint slides in the next week or so after we get everything organized. But the webinar link should go out tomorrow if you want to rewatch it or share with your team. How about?
Sorry, Brenda, may I add some things on top of Hemlin response? If you can give me maybe back I can share my screen, because I'm not sure that everyone knows what is short channel and why we need short channels. So I'm gonna share hopefully.
Yep, I see your screen.
The right one, yeah. So. So you should see it soon. Yeah, indeed. So we call short channel as we are having a really short distance between 2 optodes. So as you can see here, we are having 1 emitters and 1 receivers with 8.4 millimeters distance between both to provide short channel. Short channel aim to make the acquisition of the extracerebral hemodynamic activity only. So we are not going deep in the brain, while with the medium to long channel we are going deeper in the brain. So just to make sure that it's clear for everyone, and it is not mandatory to get this kind of short channel activity or short channel system on for any publication. The way that we are doing it and addressing that is that any of our thoughts can be tomorrow morning a short channel. So we can couple an emitters and receivers, and we can choose where we want to place them anywhere on the let's say on the device. So you can either choose to get one or a few short channel on the headset, 1, 2, 3, 4, up to a few. So it's up to you to choose which one you want to get, how much you need, and where you want to place them. Thank you.
Okay, so I'm just keep showing your screen here, or I'm just going to leave you presenter because you might want to share. You might want to share your screen again. I'm not sure. So Junji, sorry if I'm mispronouncing that, asked how heavy is the whole gig? Is it comfortable to wear?
So for the headset, you mean?
Yes.
Yeah. So actually, the fact is that, as you probably have seen, the device by itself. So if I'm coming back on the device, It has been designed to be wear on the nose support and on the back of the neck. So we can, and I did not tell you before, sorry, we can also go up to the cerebellum for acquisition. So the weight is about 700 grams for the total headset, I mean. So even, and we had several acquisition on children from 4 years old. So I mean, for 4 years old, it's fine. We can choose indeed. In any case, we can remove, let's say, the electronic box with a part of the cable to make sure that either the participant, we could be elderly or it could be a child, don't have the total weight only on the head. So we can improve that. And just for your information, in terms of comfort, I mean, I used to wear it for a 6-hour drive in Europe, and I mean for a long acquisition drive, except that having a specific design while driving my car, it was good actually to make it to see how long can I wear. So it was a 6-hour drive, so no issue with that. The point is that as we can remove again the hair below any of the optodes, what we can do is just to tighten let's say the exoskeleton device on the head to avoid moving, but we don't have to tighten too much to get signals. While in many other EEG cap based device for fNIRS, we have to tighten very intensively, I mean, on the head, trying to get access to the skin, and this could be really painful for the participant. Short question, long answer.
So since you mentioned hair, I'll go ahead and ask Elizabeth's question. What hair types have you used? How does that work for the different hair types?
So far we've been using for any hair types. Also, the more the hair are dark, the harder it is to get signals. Usually, this is due to the fNIRS modality by itself, the way that hair are absorbing the photons, and especially dark hair. That being said, we can use it in any heads. Then it's coming up depending on the way that the hairstyle, I mean, I'm not so familiar in my English, but the hairstyle, it could be a bit time. So what we are also doing to help and to try to get it easy to separate the hair, we can get a bit of water on the hair just to get it wet, so we can separate the hair below the optodes very easily. So this also helps to make sure that we can increase the signal's quality, not with the water, but just making sure that the water can go and can provide a good way to separate hairs.
Okay, I think this question is for Fraser. Does Acknowledge support ERP calculation too? This is from Frederick.
Yes, it does. Yeah, there's automated analysis in there. As long as you've got the event marks recorded from your stimulus presentation or from the task that's being performed, then we can run the automated analysis and extract the measurements or averages from those events.
New questions have come in, but I'm still going to work with the ones that came in earlier. This is from Wei Ling, also probably not pronouncing that. How is the movement artifact filtered in real time on fNIRS with MedelOpt?
So I would say that we can move. The more we move, the more we will be artifacted. We have shown with a lab back to a few months ago during human brain mapping or the fNIRS conference. We show some publication. I mean, while doing cycling tasks at 3 different tempos with very high tempos, and we have noticed that the headset does not move while doing the task. I mean, we have measured the variation in the way that we place the headset during the task, and there is less than 2% of variation the way we place it on the head. So we have some very good results comparing with let's say, movement. That being said, if we are jumping or doing very high movement, it will of course affect the signal's quality. So we can remove a part of it while doing the post-processing, of course, but is less sensitive. fNIRS is very less sensitive than EEG, of course.
So Hanley, I see your question about the motion artifact. Let me know if that answered your question or if you want me to do a follow-up. So Cork has been waiting a while. This is an interesting question. Has the device been used in veterinary medicine, pigs, other types of animals?
Okay. So this is first, let's say, a research use only device. So this is not a medical device by itself, but it can be used in hospital setting for research use through IRB in the US and in any ethical committee in Europe. So absolutely no issue. University, hospital as well, but only for research application. With regards to the participant, of course, I already emphasized can be used on child, on elderly people, on adults, on, let's say, whoever. Then babies, I mean, we can't use the exoskeleton on babies. You can only choose to use, I mean, the sensors and to place them on babies' heads. But the exoskeleton, of course, has not been designed to address babies' heads. And for, let's say, pigs, I will let you know in 2 weeks' time, we're going to have our first test in pigs in 2 weeks' time in Spain.
Okay. And I don't think it's MRI compatible, right?
No, it is not, indeed. This is LED with photodiodes, so we are not using laser. So this is not fMRI compatible. Yeah.
Okay, so I think that's it. I think that's all we have time for today. We do have, we will answer your questions in a follow-up document, and I appreciate all the questions. I'm sorry that we didn't get to them all today. I know that's it's a little disappointing, but we promise that we'll get the answers for you. Let me just show you a couple of things before we close. Whoops, not that. Hang on 1 second. Let me just get back over here. Okay, here we go. So we have the MedelOpt system on our website. You can search for it. There's a link of the mobile system. I do want to share with you that you can go to any page on our website and scroll all the way down to reach out to your local salesperson. Mine is Amy Walker because I'm in California. You can also go to our support page on our website, and you can email our support team. First, you need to have the product, and then you can email the support team, and we're happy to help you. We have a great team of support professionals that are here to help. And if you want to attend additional webinars or watch one of the 80+ webinars that we've done in the past, you can just go to our webinars page, and you can scroll down and watch any of these webinars on demand at your convenience. Now, I wanted to share about our next webinar. We have an eye tracking glasses webinar with live areas of interest. This allows for real-time views of where the participant is looking and specifically where they are focusing on your relevant areas that you've selected. So please join us on February 23rd if you're available. It's going to be a great webinar. And now I have some closing comments. As a reminder, today's webinar was recorded. We'll email you a link of the recording, the slides, and the Q&A document. When you close the webinar window, please fill out our survey. We'd love to hear back your feedback and ideas. You may visit biopac.com for additional references, including screencasts on specific features, analysis tools, application notes with step-by-step instructions, and of course, information about upcoming webinars and in-person events. We're back. All right, experts, anything else before we close? I guess.
That's good.
I think we're good. I think we covered A to Z.
All right, well, thank you, audience, for joining us and being so engaged. You had a lot of great questions, and we'll be answering those and sending out an email to you in the next couple weeks. So have a great rest of your day wherever you are. Stay warm if you're in Michigan or other frozen places in the Northeast and Europe. So take care, everyone. Have a great rest of your day. This concludes today's webinar.
Yeah. Thank you. Thank you, Brenda.
Thank you.
Bye.
Bye.